Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speakers your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Steve Tingley, VP of Sales, and Jamie Pizer, Vice President, Fluid Management, both from Repligen, and Patrick Gilbert, PhD, Director, RD&E, and Andrew Masters, Head of Business Development and Strategy, both from Purolite and Ecolab Company.
Hello and welcome. Today, I want to make the case for collaborative technology partnerships. Repligen has been in the business of protein A since the development of the first recombinant protein A molecule in 1985. In 2010, Repligen's strategy was to develop in-house a competitive agarose-based protein A affinity resin. The venture to commercialize this strategy halted because there was no clear access to two critical elements. First, access to a high-performance agarose bead, and second, access to commercial-scale GMP-ready bead manufacturing. In 2015, Purolite, a company with deep technical capability in bead technology, tried to execute the same strategic vision. The challenge for Purolite at that time was access to cutting-edge protein A ligand technology and commercial-scale GMP-ready ligand manufacturing. Two companies, the same strategic vision to improve protein A affinity purification through better performing products, better economics, and improved supply chain security. The potential for collaboration was clear. But how could it be made to work, both for the supplier companies and for the industry users? So by way of an introduction, I want to take a few minutes to set the scene for the current Repligen-Purolite collaboration. Share some thoughts and experiences of how this works from both the supply side and the user side. My goal is to briefly set the table for the technical payoff discussion that will be presented next by my colleagues. So if we take a moment and step back and look at where we've been, we can see that with respect to the industrialization of monoclonal antibodies, maybe back to 1953, affinity chromatography as we know it and talk about it today. By the 1980s, we saw the first production of therapeutic monoclonal antibodies. The first protein A resins at this time were not caustic stable and had poor capacity. So there were many technical challenges to be addressed in the coming decades. By 2012, 90% of mAb proteins are purified by protein A. At this time, protein A affinity chromatography is determined to be simple, robust, and cost-effective. At this time, modern protein A resins offer extended cycles with caustic stability and increased capacities approaching 40 mg per mil. By the time we reach the 2020s, there's one supplier that has historically controlled protein A resin supply. Why is this? Well, maybe it's because of regulatory concerns, maybe because of a lack of at-scale GMP manufacturing facilities. But whatever, what it leads to is a supply chain risk to resin supply and a lack of alternate approaches to future technology development. What are the new demands that are going to drive industrialization of monoclonal antibodies for affinity chromatography in the future? There are three concepts we ought to talk about. Firstly, the advent of new processing models. Certainly continuous processing or connected processing are key subjects of discussion these days. How can such a technical collaboration help? Well, we can offer resins that can offer the best-in-class flow and binding that support continuous or fast-flowing processes. Secondly, new purification requirements. Perhaps in the case where we have new therapeutic antibodies like bispecifics or antibody fragments that necessitate adjustments in purification methods. Again, a collaboration of this nature can result in resins that can enhance or eliminate specific binding and that can elute at higher pHs and minimize aggregation. The third leg of the stool is supply chain, particularly raw material supply chain risk, as highlighted in the recent pandemic. And there are several chromatography resins that were single-sourced and subject to very limited supply. And again, a technology collaboration of this type can create a viable alternative vendor that is innovative, capable, and reliable. The benefits of true competition are clear. So why are successful collaborative partnerships so rare? There are 4 common challenges: shared profitability, commercial control, access to partnership IP and R&D developmental control, and customer adoption risks. Purolite and Repligen addressed these challenges early. Firstly, they developed a partnership engineered to deliver a cost-effective affinity resin. Secondly, they created clear strategic focus across all affinity modalities between the partners. With respect to IP partnership and R&D control, the partners shared integrated core technologies and intellectual property such that product performance enhancements could be seen through combined R&D teams. And we'll see more of this in the next slides. And finally, and most importantly, customer adoption risks. First, the ability to have long-term agreements with renewers to secure future technology access to these products for the customers. Secondly, to integrate supply chain for ligands, beads, and finished goods of resins or pre-packed columns. The ability to offer a collaborative customer support, maximizing application expertise from both partners. And finally, partnered voice of customer that creates proactive identification of market needs to drive future innovation. At this point, in conclusion, we understand that the current vendor interaction model, a one-to-one model, one vendor, one customer, needs to adapt to a future of one customer, many vendors, so that we can apply that the customer, whether they're procurement and sourcing teams or their quality teams, have the ability to interact with more than one vendor partner. Having defined the problem, I'd now like to look a little bit further into the future and see what technical benefits have derived from these kinds of partnerships. So, how does the Purolite Repligen partnership work? Quite simply, it's best-in-class beads with best-in-class affinity ligands. They're derived from the Purolite jetted technologies or from Repligen's novel Avitide ligand libraries, which are combined into pre-packed column formats or indeed free resin for self-pack that's provided to the customers through a strategic partnership that's integrated and augments solutions for the end users. So what about examples of meaningful innovation to date from this partnership? I'm just going to touch on a few. Firstly, Presto jetted A50 with the NGL Impact A ligand. This is for traditional MABs and high productivity manufacturing. Secondly, Presto jetted A50 high pH with Repligen's NGL high pH ligand. This is for high pH elution and low aggregation of proteins. Thirdly, Presto CH170. This is a 70 micron bead with Avipure's CH1 ligand, which is a high capacity fab fragment binder. So where else might end users look for partnerships and performance enhancements? Well, if you look at the benefits that two partners bring to a collaboration, you can see it may be in dual site manufacturing for the beads, for the resins, for the ligands. It may be in large ligand libraries from Repligen's Avitide capabilities. It may be custom bead development to address specific technical challenges from Purolite's jetted technologies. It may be from pre-packed column technology from Repligen's Opus products. And finally, perhaps even single-use technologies for process management, for systems, and for CTech in terms of the ability to monitor inline real-time protein concentration management and GMP control. So in conclusion, there are plenty of opportunities to explore to find performance enhancements from such a partnership. And with that, I will pass over to my colleague, Jamie.
thanks, Steve, for the great introduction and background. As Steve mentioned, Repligen has a deep history around protein A. We were first to clone protein A in the 1980s. Over the past 20 years, we have manufactured greater than 90% of the protein A ligands used within commercial monoclonal antibody purification processes. In that time period, we set the standard for protein A development and production. Repligen has grown its technology portfolio significantly in the past years. Other areas include pre-packed columns and upstream intensification all the way to analytics. Affinity chromatography has always been in our core DNA, though. In 2021, we made significant investments in our affinity ligand business with the acquisition of Avitide. Avitide is a leading affinity discovery and development company located in Lebanon, New Hampshire, very close to Dartmouth College. Avitide has built a technology platform around a diversity of affinity ligand scaffolds and libraries for each of these diverse scaffolds. The foundation of this platform is built around highly specific affinity binders that are stable, also capable of providing high binding capacities, and all within rapid development times. Over the last decade, affinity programs have spanned across all kinds of different therapeutic molecules: proteins, enzymes, blood factors, hormones, viral vectors, including AAV, vaccines, and so on. Greater than 30 development programs have been completed with 20 plus partners or customers with a very high success rate. Today we are talking about partnerships. The Repligen Purolite partnership is not a new one. We started this journey nearly a decade ago. At that time, Purolite had a vision to establish a world-class agarose bead capability. With Repligen's history and capabilities around protein A manufacturing and supply, this made perfect sense for us to combine forces. Early on, the conversation was mostly around protein A. As the partnership matured, the conversation turned to what other things can we work on together? What other needs are there currently in the market? How can we develop solutions that add value to the development and manufacturing communities? So what was the market saying? There are fewer standard FC-based molecules being developed, such as standard monoclonal antibodies. Molecules are more complex. They can be multivalent, contain novel fusion constructs. These molecules do not behave as well as standard mAbs. They have increased potential for aggregation. They might be more sensitive to low pH elution, which becomes problematic with standard protein A elution conditions. The narrow standard window for process operation is another factor, and capacity is king and directly tied to process economics. CIP sanitization is important. Hydroxide stability is a must. This led us to develop the product that we'll discuss in greater detail today. A protein A resin that still retains all of its must-have properties, but can also operate under mild elution conditions. So for this specific program, Repligen partnered with Navigo. Navigo is a protein engineering company located in Germany, who had built a strong technology around the engineering of protein A. Their unique technology uses a shuffling approach and directed evolution. Protein A, as a wild-type molecule, contains 5 domains: E, A, D, B, C. Each of these domains are very similar in structure, but do contain differences. So by combining the best of each domain, you're able to assemble the most optimized domain. So in this cartoon, which is kind of fun with the cars, you'll see cars a different color. So by taking the top of the car or the doors, the hood, and shuffling together, you come up with the car that is most appealing to you or most optimal. And then after that, you can do other fun things like add racing stripes, T-tops, whatever you want. So those really represent individual mutations to get you at your endpoint. So by doing this type of optimization approach, we can really look to strengthen the sodium hydroxide stability, change specificities, and at the end of the day, for this project, the goal was to really increase the elution pH or make it more mild. So while doing that, we also wanted to maintain sodium hydroxide stability, make sure that the capacity when immobilized is still really good and equivalent to the other products that we've made in the past. So from that point, we're able to end on a molecule with all of these properties. Now I'm going to hand it over to Patrick, who will highlight the product.
Thank you, Jamie. So now I'm going to specifically talk about Presto Jetted A50 High pH. This product combines Purolite's proprietary and patented jetting technology, which is used to manufacture Liagro's beads in a more monodispersed fashion compared to traditional manufacturing methods used for resin production. And a novel protein A ligand from Repligen, which utilizes the advanced ligand screening approach mentioned previously to produce a unique and novel protein A purification solution, which has been specifically designed for FC-containing molecule, which enables significantly higher elution pHs to be utilized compared to what is commercially available. This enables higher recovery yields for pH-sensitive molecules, specifically for bispecifics, FC fusions, and other non-standard antibodies that are prone to precipitation and aggregation, while maintaining the critical processing parameters required for effective utilization. This has the advantage of being almost a direct replacement for a standard protein A resin and enables a higher operating window to be utilized while maintaining a relatively high capacity resin. So for Jetted A50 High pH, we screened multiple ligands in partnership with Repligen. On the graphs shown on this slide, there are two of the lead candidates of the high pH with different genetic sequences and slightly different behaviors. And our leading commercial protein A resin, Presto Jetted A50, and the market leading competitor. So when we compare a linear pH gradient with an FC fusion protein from pH 3 to 4, the standard protein A resins elute off at pH 3.4 to 3.6. Now, the lead candidates had much higher elution recoveries at 4.8 and 5.2. Now, when we compare the different ligand constructs against a wide range of antibodies, you can see slightly different behavior between the candidates. So on one of them, there is a key, the ligand is clearly influenced by VH3 binding, and one of them was not. So the ligand we commercialized to put on high pH resin was ligand number 2, which was then not influenced by VH3 binding. And this allows it to be used universally across antibodies without having to worry about unwanted VH3 binding, because this then would create a stronger bind and then cause the protein to stick at the mild pH conditions and would then have to be eluted off. So utilizing the ligand screening approach, we zoomed in on the final candidate based on a scientific approach. What this means for the end user is a high capacity resin that can achieve 60 grams per liter binding capacity at relatively short residence time, has excellent mass transport effects due to the jetting technology, and has good alkaline stability, so standard CIP conditions can be used to clean the resin in between cycles. So in-house, historically for QC test and release, we use polyclonal IgG, and that binding capacity is shown on the graph on the left. And the data on the right is with an IgG1 feed, and the binding capacities are comparable between the polyclonal and the monoclonal. So effectively, we have made a market-leading novel resin that is first on the market to be able to be used to elute an antibody up to pH 5. Now I am going to hand it over to my colleague Andy, who will walk through this innovation in more detail.
So thanks to Jamie and to Pat for giving us an overview of the development and the functional properties of Jetted A50 high pH. Now I'm going to focus on the use of Jetted A50 high pH in practice. And I'm going to share with you a case study from one of our customers, that's Kayla Barry from Maplex International, who presented this during a conference in BPI Boston, September of 2023. She presented the study around the use of Jetted A50 high pH resin compared to the use of a standard protein resin for the purification of 3 bispecifics, which they had in-house. For those of you who don't know who Maplex are, they provide global contract research and contract manufacturing services in development and manufacture of biopharmaceuticals. So Maplex's first interest in Jetted A50 high pH came from the challenges that they'd experienced in-house with bispecific purity when using standard protein A resins. Being a CMO, Maplex had assessed manufacturability for several bispecific antibodies, and what they would observe is that for some of the bispecific antibodies, they tended to become unstable during standard protein A elution conditions, typically between the range of 3.5 to 3.7. And what they found was that this can cause aggregation and drive fragmentation. One of the most difficult molecules that they've investigated is an antibody that they call BSABZ. It was particularly challenging. It has major issues with aggregation and fragmentation, and it made it really difficult to purify. Quite often, they would only end up with 60% monomer remaining after the protein A step using these typical elution conditions from protein A. The graph here shows a chromatogram on the top right, and on the bottom left, it shows the actual fractions collected across the peak. The chromatogram on the top right is an isocratic elution of protein A resin. And as you can see, the red line, it has a pretty tight isocratic peak. However, if you look at the actual fractions collected across that peak, and you look at some of the contaminants and the antibody within the fractions, so you have the monomer in green, the cumulative monomer in purple, high molecular weight and low molecular weight in red and yellow respectively. You can see that for the monomeric, which is in green, it starts quite low and it really never gets above 80%, whereas the actual cumulative monomer, which is the purple line, you will see it really gets about 65% overall. For the high molecular weight, it starts at 40 and it really co-elutes across a lot of the chromatography peak, leaving a lot of undesirable material and poor resolution between this and the target monomer. So due to such low monomeric purity of the protein A step, it required subsequent polishing, again with low yield chromatography steps, and overall the product quality wasn't good, the monomer content was low, and they didn't have a lot of material to do further characterization and analysis. So Maplex were then made aware of Presto Jetted A50 by our Purolite representatives in San Diego, and they were really keen to evaluate this for the current and past challenges that they'd seen with these bispecifics in-house. They were really hopeful that their more acidic labile bispecifics could benefit from the ability to elute under more gentle conditions up to pH 5. And they looked at this really difficult molecule, BSABZ, and they purified this over a small-scale high pH column, utilizing the high pH elution that you can get with this resin. The goal was to try and prevent aggregation, achieve better separation between the high molecular weight and the other undesirable contaminants that they were observing under standard protein A conditions. 2 Purolite resins were compared in the following slides. Presto Jetted A50 and Presto Jetted A50 high pH. Presto Jet 50 will be referred to as standard protein A in this presentation, and it binds to molecules containing the FC and VH3 regions of an IgG, whereas for Presto Jet high pH, the ligand is engineered to bind mainly to the FC region with absolute negligible interaction to the VH3. So the first results that we obtained, this is shown in this slide here for Jet 50 high pH, and this was capture of the BSAB-Z antibody followed by a gradient elution. So again, we have the chromatogram in the top right-hand corner, and we have the fractions in the bottom left. If you look at the top, you can see that Mabplex packed a column in-house, 3.42 mils, and they used a slightly lower load due to sample availability. They also carried out a high wash after the load, which you can see on the chromatogram. There was a high salt wash to remove a lot of the low molecular weight species before they carried out the elution with a pH gradient, which they ran between pH 5 and 3.5. If you look at the fractions on the left-hand side, you can see that the monomer content was pretty good across the board, about 96% cumulative monomer, and they only saw some high molecular weight contaminants appearing towards the end of the gradient, so in the later fractions and the lower pH range. This was really promising data for Mabplex, since it supported the initial finding, that eluting at lower pH was causing them aggregation issues for their standard protein A resin, and it gave Mabplex the potential to develop and optimize a high-yield isocratic elution of BSAB-Z using this Jet 50 high pH resin. However, unfortunately, in this case, due to the lack of additional feed material for BSAB-Z, it really wasn't possible. Therefore, Mabplex decided to widen the evaluation to look at other bispecific molecules of similar design that they had access to and were available in-house. So Mabplex looked at two different additional molecules. They called them BSAB-X and BSAB-Y. BSAB-X was mostly stable with some aggregation and fragmentation, whereas BSAB-Y, again, when using standard protein A resins, they did see a lot of aggregation and fragmentation. For the analysis for this study, they only had size exclusion analysis available. However, for future work with Mabplex tend to carry out with a high pH resin, they want to look more at the host cell protein profiles, capillary electrophoresis, and in-depth more analysis to really see some more benefits of the high pH resin. For this study, the main focus for Mabplex was high molecular weight. This is their biggest concern because they find these harder to remove downstream. Low molecular weight species was less of a concern since Mabplex typically can remove these by an optimized wash step. Compared to this BSAB-Z, both of these molecules behave a little bit better compared to that molecule. However, what they wanted to see was, compare the purification of these two bispecifics X and Y using the Jet 50 high pH resin against, the standard in-house protein A resin of Jet 50. So they wanted to compare this and see what would happen if they eluted these with a pH gradient in the region of 5.3 down to 3, compared to the standard elution conditions of 3.5 to 3.7. What would be the difference in purity of these molecules? The method that they used was relatively standard for protein A. However, when you're actually using Jet 50 high pH, you have to be careful when you're introducing some low pH washes prior to the main elution. You have to be careful that you're not getting too close to where the antibody elutes. So it's a consideration that you'd have to bear in mind if you're just doing a straight swap between the conditions that you might have used on standard protein A. In this case, they used pre-pack columns, 5 mil pre-pack columns from Repligen, the mini-chrom columns, for both the Jet 50 high capacity standard protein A and the Jet 50 high pH resin. The loads, they kept the same as what they'd used for the BSAB-Z molecule, 20 mg per mil, and they collected 1/3 column volume fractions using a watch command to collect the peak with a normalized path length 2.2 centimeters. The gradient was from pH 5.8 to 3.3, and it was carried out over 20 column volumes. It doesn't mention which buffers they used in the slide here, but if you want to make a pH gradient, Purolite would typically recommend using something like 50 millimolar citrate pH 6 to 3, using that type of buffer. So this chromatogram is the results for bispecific antibody X, and it highlights the difference between the different protein A's ran with the gradient from pH 5.8 to 3.3. The blue line is PrestoJet A50 high pH protein A, and overlaid on top is the red curve from the PrestoJet A50 or standard protein A. For the blue curve, you can see that the elution started for high pH around about 71%, so it's closer to the higher end of the pH gradient, and you can clearly see some visible separation in the shape of the UV peak. For PrestoJet A50, the elution begins around 86%, so towards the lower end of the pH gradient, and there's no real resolution, and you get a very tight elution peak. So if we look at the size exclusion analysis of the fractions and the cumulative monomer content, and on the graph what you'll see is the results for high pH on the left and the results for the standard protein A on the right-hand side. You can see that for the Jet A50 high pH on the left, a cumulative monomer content of 90% was achieved, with the highest fraction containing over 93% of the monomer species. The remaining low molecular weight species, which is the red line, which is the yellow line, eluted quite early on in the gradient, and the high molecular weight species were pretty consistent across the whole of the fractions collected. For Jet A50, which is on the right-hand side, you can see the cumulative monomer content was about 84%, with no fraction going above 87%. So all of the species co-eluted at consistent ratios, so it was quite far away from their preferred ideal goal of trying to get about 90–95% monomer purity levels to do the further analysis and characterization. As I mentioned, for high molecular weight species for the jetted high pH, they were consistent across the board, and it was the same for the standard protein A. However, Maplex could conclude again, for future work, the high pH elution trend was really showing promise for them to be able to optimize isocratic conditions more easily to maximize their yields and to achieve a higher monomer content capture for the protein A step than typically that they would achieve with standard protein A. So for bispecific Y, the species separations followed a similar trend to bispecific X. However, for bispecific Y, using the Jet A50 high pH, you can actually see even more resolution between the two main species during the pH gradient compared to that obtained using the standard protein A, which was the red line on the graph. Jet A50, although there was some separation, it was minimal and there was only a shoulder. However, once again, the elution of the antibody on the high pH resin comes off much earlier, higher pH, around 38% buffer B, and for Jet A50, it came off much later, around 86%. So again, it's reflecting the higher and lower pH elutions respectively. If we look at the actual protein A fractions that were obtained, again, on the left-hand side, high pH, and the right, standard protein A, you can see that for the size exclusion results for BSABY, the cumulative monomer level was about 96%, which is very high, and again, was shown more of an increase in the high molecular weight species as you get towards the later fractions or the lower pH. And for the Jet A50, which is on the right-hand side, although the monomer levels were high, what was quite striking from this was that the high molecular weight species significantly increased towards the later fractions, up to about 30%, and it resulted in a lot more co-elution due to the overall elution pH being much lower than the high pH resin. Maplex were really pleased with the results, and they could see these potential benefits of Jet A50 high pH in order to purify these challenging molecules. They could see that they could do this more easily with minimal optimization or messing around with buffer additives. And this would allow them to purify products much closer to their specification window of yield and monomer that they require for further analysis and characterization, and obviously being a CMO for subsequent scale-up. So in conclusion, PrestoJet A50 demonstrated clear purification benefits for the bispecific molecules evaluated. They understand the protein A conditions for many of those bispecifics. They found that it agreed with what they were thinking, that low pH conditions on a standard protein A resin can actually drive conditions towards aggregation and co-elution of monomer with undesirable species. And this really causes them problems further downstream. It's much more difficult to separate the high molecular weight from the monomer in subsequent purification steps rather than doing it on the capture step. So for the molecules that Maplex evaluated, high pH demonstrated overall improved separation and recovery of the monomer species. And it was really great for Maplex. These early data points pointed to the fact that they could use a novel protein A resin potentially as a platform solution for biospecifics, which could help them sort of expedite the purification to get these molecules in required quantities and the specification for further development, investigation and analysis. And they're really keen to follow this work up. They're looking to evaluate this further with other biospecifics, but take it more and have a look more at things like host cell protein and DNA clearance as part of this ongoing evaluation. So it's not only Maplex who have given us some great feedback regarding Jet A50 high pH and some of the performance benefits. But we've heard from a number of discovery labs where many molecules are screened and purified at small scale for early analysis or characterization, that the use of high pH resin resulted in a very high monomer content, removing the need for other purification steps such as small scale size exclusion. And that simplified for them and speeded up the purification of many different molecules. In our own labs and from collaborators, early collaborators such as Alvatech, we have seen significant reduction in host cell protein and recombinant DNA in the elution pool from the high pH protein A column as a result of this higher pH operating window. From multiple evaluations of customers and feedback, including Maplex, we found fewer aggregates and the ability to increase monomer content and overall recovery without the need to use additives in the elution of the protein A column. And for one CMO, we found that when they were trying to apply a customer or their own platform for protein A, they were very low recoveries, which meant that by using high pH resin, they could rectify this situation quickly and then speed up the development and transition of that project to manufacturing. So now I'm going to talk about the next innovation. And as mentioned early in the presentation, Purolite and Repligen are looking to continue to expand a toolbox of affinity resins, which can facilitate the purification of these more challenging antibody variants. And as part of that, in February of this year, Purolite launched Presto CH170. The Presto CH1 resin has a ligand which is designed specifically to bind with a high affinity to the CH1 domain of human IgG and related molecules. And I think all of us on this call are aware of the increasing trend of these more complex antibody-based therapeutics coming to market. So having a wider range of affinity resins targeting different regions of the IgG molecule can help simplify and speed up the purification process. I think as a PD scientist, having wider availability of selectivities that can bind to the different regions of the antibody will facilitate, an optimal purification against their intended target. I think we're seeing what doesn't bind to a resin as important as what does bind to it, because there are closely related contaminants which obviously make the subsequent purification and the polishing steps much more difficult if they're not removed as part of the capture step. The selection of the base bead and the coupling of the ligand have been engineered for this molecule to deliver, for this resin to deliver really high productivity and make this a resin that's really can be used for the GMP manufacture of antibody therapeutics. As mentioned, this resin was designed to facilitate the purification of antibody-based molecules where perhaps binding via the FC region of protein A or other purification methods, or resins which bind to the variable and constant regions of the Fab light chain, for example, such as protein L. These can often leave these difficult to remove contaminants such as overexpressed light chain or homodimers of the target molecule. And having the ability to select an affinity resin, which removes as many contaminants as possible during the capture step, has the subsequent knock-on effect of reducing the number of unit operations and simplifying the subsequent polishing steps and maximizing the overall yield. To make Presto CH1 a viable option for GMP manufacture, this was coupled to a 70 micron bead, which delivers a high dynamic binding capacity, 40 grams per litre, which I'll show you in a moment. And it also has excellent pressure flow characteristics. It's not shown on this slide, but you should be able to expect to get 300 to 350 centimetres per hour in a 30 centimetre diameter column with a 20 centimetre bed height. And if you couple this with the increased resistance to sodium hydroxide for cleaning in place, this results in a resin which is going to deliver improved process economics over resins with similar selectivity. If you don't know about Purolite's resins, all of Purolite's resins, affinity and ion exchange, are made using jetted technology, which from a manufacturing perspective is faster in our hands than traditional emulsified methods, and it also reduces the use of solvent and lowers production waste. The process, as Pat mentioned earlier on, of using jetted agarose is that we end up with a higher degree of uniformity around the target bead, which in this case is 70 micron, and that supports the ease of packing whilst delivering high efficiency, fast mass transfer, and really high capacities over a range of residence times. Which is shown on this graph here. This actually shows a dynamic binding capacity, 10% breakthrough curve for a monoclonal antibody, IgG1, which is shown on the left-hand side of the graph. And it shows the capacity that you can achieve for an IgG1 on the CH1 resin across a range of different residence times. So typically, if you look at 4 to 6 minutes, you should be able to attain 40 grams per litre of MAB binding per mil of resin. For fabs, we have also seen capacities above 35 grams per litre, as shown on the right-hand side of the slide, which is significantly higher than we observed during benchmarking with a commercial resin, which has similar selectivity for CH1. And having this higher dynamic binding capacity supports the use in a GMP manufacturing process where high productivity and favourable economics will always be in focus. So also contributing to the overall economics is the stability of the resin to sodium hydroxide for cleaning in place. And this is what this slide shows. It's a expedited study where you basically expose the resin over a period of time to different concentrations of sodium hydroxide and then periodically test its dynamic binding capacity and compare it to the original capacity before the resin was introduced to the sodium hydroxide. And if you look at it, you can look at 90% and 80%, and those are the values where people will typically build in a safety margin for loading onto a chromatography column, and you can get an indication of the typical resistance and potential lifetime exposure without exposure to representative feed. And that's really the caveat of this study. It's not carried out with representative feed. From time to time, you will do periodic testing of the DBC and you use pure IgG. So the actual number of cycles obtained will very much depend upon the feed, the concentration of sodium hydroxide used, and the contact time that you employ as part of your CIP strategy. We have ongoing many evaluations of lifetime with different feeds. However, we also have representative data, which was produced during the development of this resin, kindly provided by Repligen here, where we used a IgG feed, which was depleted by passing the IgG over a protein A column. It removed the antibody and left depleted CCF, which was then spiked with 2 mg per mil of fab and loaded up to 40 mg per mil capacity in this instance. The column was then ran for up to 50 cycles and cleaned with 0.1 molar sodium hydroxide with a 15-minute residence time every cycle. And the yields and levels of contaminants, as you can see from the slide here, were consistent against the target of 50 cycles. The remaining dynamic binding capacity, not shown on the slide, stayed above the 80% of that obtained at the 10% breakthrough. So we have Presto 70 CH1, which has been added to our growing Affinity toolbox. We don't intend to stop here. This is continued development between Repligen and Purolite to expand the availability of different affinity selectivities that will help overcome some of the purification challenges we're seeing with these modern antibodies, and the requirement to remove contaminants which have similar structural properties. So we have a number of ligands in the pipeline, which we will commercialize over the next few years. But alongside that, we're trying to take what we've learned from our existing ligand design and technology and apply it to future products. So as a leader, in ligand and bead design, we're working really closely to build on this knowledge. For example, we've learned from the high pH how to design in less aggressive elution conditions, and we want to apply that to future protein A ligands. We also want to look to increase the stability to cleaning and sanitization reagents, and also look to see whether we can make them more resistant to proteases, for example, which can occur more often in feeds from high density cell lines. Alongside that, we're looking to the future. We know the antibody market will change and trends will change. So we're always looking to identify and develop new ligands. which can facilitate the purification of future antibody-based therapeutic formats. And we really do, in this regard, appreciate feedback, and we encourage our clients to reach out to us to discuss their current challenges, open collaborations, and continue to drive this innovation. And with that, I will pass to the audience poll.
So I just sent a survey to the audience that we kindly ask for your participation in. The 2 questions read, how do you see strategic partnerships benefiting the industry? And what current and future gaps do you see for affinity purification tools? So if you could just take a moment or two to leave an answer for those 2 questions, your participation would be greatly appreciated. And then we will get started with Q&A. We'll just give a few moments to for you all to answer those questions. The questions won't be shared with anyone else. They are private. All right. Well, thank you for your participation and answering those couple of questions. And I think we can begin the Q&A now. So as a reminder to our audience, you can go ahead and continue to type in any questions you have. And our first question is, in what ways do you consider the industry will have to operate differently to take advantage of such product collaborations?
I guess I'll take that, Alex. The industry, has been used to working over very many decades quite successfully in a one-to-one model, right? Where there's one party responsible for developing, commercializing a product, and one party that's responsible for utilizing that product. And I think what we're suggesting, as we move forward and we do these combination products, is that we're going to have to be used to more of a one-to-many model. And I can give you some examples where we've seen this. One comes up in the area of product supply, procurement, and paper workflow, whether it's orders or whether it's documentation for incoming use in the warehouse. How do we get the appropriate information from 2 separate parties? Clearly some of this can be integrated, but others of it may have to be coming from 2 separate parties. There's another area which usually brings up questions, and that's around the questions of quality and product performance. Who's responsible? Who has the ownership? What are the practices going to be as we onboard collaborative technology projects? Particularly as it's related to qualification document, maybe complaints and scar closure. But these I think are all very practical issues that can be resolved, but they need to be resolved early. So where we've seen the greatest success is when this is realized early, and that one of these collaborative development projects is started within a user community, and they early get the procurement teams and the quality teams involved so they can understand the processes, and then they can adapt the programs that they have in place to enable them to take advantage of the benefit of some of these joint technologies. Thank you.
That's great. Thank you, Steve. And our next question is, what flow rates would you typically recommend using with high pH?
I will take this one. So that's a good question. For high pH, we would recommend in a 20 centimeter bed height to run the column about 200 to 250 centimeters per hour maximum flow rate. To be able to, for quicker running, we would recommend a shorter bed height, and that will take advantage of some of the other process economics by utilizing this resin, such as increasing productivity while reducing buffer consumption.
Awesome. Thank you, Patrick. And our next question is, did Mabplex consider using high pH as a platform resin?
I can take that one. When I spoke to the lady who did the presentation in September, for their application, they definitely are considering it because they generally have a lot of molecules where they are expecting to see some sort of instability, aggregation, and fragmentation. So I think for them, it's really expediting the purification. They don't have to mess around too much with trying to optimize it. They're already being able to use gentle conditions to reduce this happening. So yeah, they were looking at it. I guess if you wanted a high capacity and you didn't have stability issues, then Jetted A50 would be our recommendation.
Awesome, thank you. And our next question is, in the case of a collaboration product, what should a buyer's expectation of quality, technical support, and availability be?
Yeah, I can take that, Alex. And I think the point here, and really a large point of this whole conversation, is that if we really want technology advancements, we're going to have to expect to work in collaboration partnerships. But we shouldn't be afraid of that, because the answer to this question is exactly the same as for every other commercial process. The standard has been set in the industry for commercialized products, and these are no different. So the quality should be the same. The technical support will be the same. The responsiveness in terms of availability, manufacturing information, RSFs will all be the same. The difference is that there will now be two of everything instead of one. So in this case, two manufacturing operations, one for the bead, one for the ligands, and another one, in fact, obviously for the immobilization and the resin manufacture. So there are two quality organizations, two R&D organizations, two application organizations, and two commercial organizations. But what you need to do early on is to clearly define a precise contact and how individual end users get the support they need. So this is done by setting up a single exclusive supply strategy. Primary point of contact. In this case, it's Purolite. But if there are issues and questions and opportunities that can be resolved by engaging with the Repligen technology teams, a little bit like this joint webinar. When the occasion demands, you will see representatives from each of the companies involved with core technology. Thank you.
That's great. Thank you, Steve. And our next question is there an assay available for CH1?
There is indeed. So the ELISA assay for CH1 can be purchased directly from our friends at Repligen.
Terrific. And our next question is, at what scale have you packed Presto Jetted A50 high pH?
I can take that one. I can see two questions similar with regarding the A50 bead. Obviously, Jetted A50 and high pH are both constructed on the same bead. So from the A50 perspective, we've packed 1.6 meter diameter columns with 17 centimeter bed heights, 1 meter column with 20 centimeter bed height, and in-house a 0.6 meter column with a 12 centimeter bed height. And that's what people are using for smart cycling, where they take advantage of the fast mass transfer, the high capacities and shorter residence times to carry out process intensification. So they're both on the same bead. We expect no problems at the moment. We're probably looking at high pH is only being launched for 2 years. So we're seeing people with like 45 centimeter pre-pack columns for sort of phase 1, phase 2.
All right. Well, it looks like we are running just a bit short on time, so if we don't get to your questions, we'll be passing them along to our speakers who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along.
I can see one that I could answer, if that's okay. There's a question around the elution volume for high pH and what are the wholesale
that was the last one. That was the last one I was going to ask. Yeah, please go ahead. Please go ahead.
It obviously depends upon the antibody. So what we recommend when you're evaluating high pHs to run the pH gradients, and then select the pH, which obviously gives you the highest pH to obtain the elution. When we did a lot of prototype work with a lot of evaluations with a high pH resin, you will see at some point that the elution volume starts to get bigger, especially if you really try and push it up to like pH 5. So, it will start to broaden. But generally, with a high pH up to 4.5, 4.7, it's no different to what you would see typically with a classic protein A resin, because it's been engineered to have a specifically gentler binding, if you like, to the actual MAB. So there's not that much impact on your facility fit potentially, or your fractions that you need to collect. In terms of the whole cell protein, we've got data that we worked with a contract manufacturer who did some evaluations with the whole cell proteins being significantly reduced due to the actual high pH elution. And that's a trend that we saw right from the very beginning. Not only whole cell proteins clearance improving due to the high pH, but also recombinant DNA being a lot lower in the eluent from the high pH column.
All right, well, thank you so much for that. And thank you to Steve, Jamie, Patrick, and Andrew. Thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you. Bye.